Well, thank you all for having me. I'm going to assume everybody can see my screen okay. So today I'm going to be giving what is really a kind of hybrid of two talks. I'm first going to talk a little bit about the recent history that's gone on in sociogenomics. I think that's just a useful primer to the field, as I think it'll be a field that sees real growth in the coming years. And then I'm going to talk specifically about a project that has a somewhat novel approach for examining gene by environment interactions. So I like to start by giving a disclaimer about what I think genetics might be useful for. In particular, I really think that genetics should be thought of as a potential candidate mechanism for explaining how people who start in similar situations end up different. And it's terrible, in my opinion, for the most part, for explaining why people who start in radically different situations don't end up the same. I think that's super important. It is not going to be a good use of genetics to try to use it to explain why people who come from very different places, have very different resources, don't end up the same. That's, I think, a fool's errand. And a lot of social science is focused on the structural forces that lead to unequal distribution of opportunity. That's what creates those radically different starting points. And I think that's rightly so. I think that that scholarship is incredibly important, and nothing that I'm going to say today is really going to change that. What I'm going to say is largely orthogonal to that. I do think there's also more to the story of human behavior. And if we really want to understand human behavior, we're going to have to grapple with things like genetics. If it was only environments that mattered, siblings would be way more similar than they happen to be. And I think this is a potential kind of motivating reason for why we would care about genetics, even though there are other things that matter greatly. In particular, if we could better understand why siblings or otherwise similar people differed, that might, tell us about developmental cascades or otherwise offer insight into opportunities for more effective interventions, or to further refine our notions about the relevant environmental impacts that are affecting the things we care about. So again, I think it's really important for us to kind of be on the same page about when genetics might be useful, when it won't be useful, and what it is that we're actually hoping to do with it. And this is my attempt to kind of at least tell you how I think about that. So with that caveat in mind, let's first start with a very little bit of biology. So the thing that I study is what is known as a single nucleotide polymorphism, or a SNP. So a SNP is a single letter change in the DNA sequence. You can see I have two pieces of DNA here. At most base pair locations across these two strands, the two pieces of DNA are identical. But at one point, we have a bit of variation, right? These two base pairs are not the same. This single letter change here is what is known as a SNP. And there are millions of SNPs in human DNA. And it's important to note that a SNP is not a gene. A gene is a large region of DNA or something much more complicated, whereas here we just have variation at a single point. And SNPs have been widely used in genome-wide association studies to build maps that tell us what parts of the human genome are related to different traits. Here is such a map, and this is based on a very brute force approach of analyzing association between SNPs and phenotypes. Basically, what you do is you take a phenotype and then regress that phenotype on a large number of individuals' genetic data. So you run millions of regressions, one regression for each SNP. And you end up with outputs like this. So this is what's known as a Manhattan plot. These kind of blue buildings are meant to be reminiscent of the Manhattan skyline. And this comes from a GWAS looking at Alzheimer's disease. And you can see, for example, there's a very pronounced peak here. This is the APOE gene that has been tied to Alzheimer's disease and related dementias in a variety of studies. It's a very kind of biologically important bit of genetic variation that is seeing a great deal of scrutiny at this point. And such GWAS have been used very widely to better understand the ways in which genetic variation is tied to variation in a variety of biomedical outcomes. So we've been doing this for about 10 years, and it's been quite a success. There's a really nice paper that I'm showing you there that discusses what we've learned over the past decade from GWAS. And it's really transforming a variety of biomedical disciplines. But there's nothing really to stop us from taking this out of the biomedical realm, right? Like all I need is an outcome and genetic data, and we have outcomes above and beyond, heart disease or Alzheimer's disease. We have other kinds of outcomes that you can imagine playing the same game with. And in fact, that's what began to happen. So as a point of reference, here is a GWAS done for educational attainment, right? A very different kind of outcome. This was big news when it came out. You can see the write-up here from the New York Times that covered this paper. This is not a paper that I was involved in. I'm not advertising my own work here. I'm just noting that this was a big prominent finding. This is again using the exact same approach that was designed to study genetic variation and kind of disease outcomes or anthropometric traits now being applied to what is a social outcome. One thing you can note looking at this Manhattan plot has to do with this dashed line. This is the line that tells us about the corrected level for statistical significance, right? We've done millions of tests. We need to correct for that fact. And what you can see is a ton of dots above that line, right? These dots all represent SNPs that show some association with educational attainment, even after correcting for multiple testing. And the basic story here is just that we have stuff from across the genome being associated with educational attainment. And this has come to be known as the fourth law of behavior genetics, which, as stated at the top, is that a typical human behavioral trait is associated with very many genetic variants, each of which accounts for only a very small percentage of the behavioral variability. So this presents a bit of a challenge, right? We don't have one very sharp predictor here. The signal is quite diffuse, and this is going to make it complicated if we want to do things like include genetic information in our models, right? So what might we do? And the answer that people have, one answer that people have started to settle on is what is known as a polygenic score. And the basic idea is just to create a weighted predictor where we use the GWAS weights that tell us about how influential a SNP is for a phenotype. And we kind of weight the genotype of an individual. And we use this kind of weighting algorithm to construct nice, continuously distributed indicators of genetic risk for some outcome. I like to think of these like credit scores. So a credit score is a single number summary meant to quantify an individual's financial risk. And here what we're doing is creating a single number summary that is meant to quantify your genetic risk for something, right? It's going to depend upon what the weights were programmed to predict. And I just think it's worth emphasizing that this is all just something we can do based on saliva, right? We take the saliva of individuals, and now using the large database of GWAS results that we have, we can construct, levels of risk for that individual without any other information about them that might tell us something about, their kind of biomedical future or any other number of things that might happen throughout the life course. So to give you a bit of a sense for how these things work in practice, I pulled a figure from this paper that was the GWAS of educational attainment. And it turns out the polygenic score designed to predict educational attainment predicts about 10% of variation in years of education. That's what's shown here in this red box. Now, how big is that? Well, I think the best way to think about that is that it's roughly on par with the level of prediction we get from either parental education or individual kind of cognitive functioning, in particular tests of verbal ability that are available in these two data sets. And so I think this is both kind of expected in the sense that individual cognitive functioning and parental levels of education are not orthogonal to what we know about an individual's genotype, but it's also, I think, very suggestive that these are fairly potent predictors, given that they're predicting about as well as what we would get with pretty decent measures from sociological or psychological surveys, right? So I think this suggests that these things might have some real heft to them. Another thing that you might worry about is that they're purely a signature of social class, of the social class that a person's family resides in. And one thing you can do to maybe rule that out is to look at siblings. So the reason that two siblings have different levels of education tend to be quite different as compared to two unrelated individuals. And so when you compare siblings, you're able to rule out confounding forces that don't vary within family. And we did that work in this paper here, where we're basically comparing predictions amongst when we kind of consider people to be unrelated versus when we incorporate family information, which are these light blue bars. You do generally see that you can predict sibling differences, right? That is, the genetic differences between siblings are predictive of later phenotypic differences, in particular differences in educational attainment or occupational attainment or wealth. There is some degree of attenuation, right? That the light bars are a little bit retracted from the dark bars. That's important. I think that relates to the genetic nurture problem that I'm going to talk about later. But in general, you still can kind of get some predictive power out of these within family deployments of polygenic scores. And so that's very important. Okay, so now what? So I've introduced this novel predictor based on genetics. What are we going to do with it? And I think this is an important question, given that there are a ton of such data coming online. AdHealth, HRS, and the Wisconsin longitudinal study are all longitudinal cohort studies that I'm guessing you all are familiar with. They have all added genetic data in the recent past, and particularly they've added polygenic scores. There are catalogs coming online that allow you to download polygenic scores for even further data sets. Polygenic scores and GWAS type data can be cheaply added to just about any data set at this point. There are people interested in incorporating this information into clinical care. So this is a clinical trial looking at heart disease and other things, and whether you can use polygenic scores to stratify treatment or further improve clinical practice. So these are poised to be a very big deal. They're I think going to impact a lot of science and probably a lot of other things that we're dealing with. And so it's timely to think through what we're going to do with them. Now, there are a lot of potential answers to this question. I have accumulated here a bunch of papers that I have learned from over the years. The two underlined ones both have some kind of Princeton ties. And so while I would encourage you to look at these papers, I also want to offer a thumbnail summary of what I see as being the precipice upon which we stand. I think there are both opportunities, in particular opportunities for genetics to throw novel light on old problems, and then also reasons for caution in both application and interpretation of genetic findings. So I'm going to talk a little bit about both of these two bullet points here with a real dramatic overemphasis on work that I was involved in. I just want to, be upfront about that fact. So the point of opportunity that I think we have is that genetic associations may help us to interpret social change, given that genes are static. And this is a really important fact, that genes are static. And so let me give you first some examples of how we might do this, and then I'll return to the fact that genes are static and what exactly that helps with. So two examples. So the first comes from a study of the effect of spousal death on levels of depression in the health and retirement study. This is a picture that has no genetics in it. This just shows you time from spousal death, whereas the negative numbers means before spousal death, the positive numbers means time after. And you can see that, when we interview people in the HRS right after their spouses died, they show, as one might expect, quite elevated levels of depression relative to what we observed previously. But those levels, decline over the subsequent year or so, and that's important. And so this is the kind of picture that we don't need any genetics for. But the question now is whether or not we can further understand who is at elevated risk for depression in the wake of spousal death if we do have genetics. And the answer is that we can. So what we can do is we can kind of take genetics and further decompose this figure into this one, where we're now able to look at people that have heightened levels of risk based on their genetics for depression versus lower levels, and see that those at heightened levels of genetic risk do in fact see a quite pronounced upward shift in the level of depression that they experience in the wake of spousal death relative to their peers, right? And this is, in our kind of conception, a fairly exogenous shock, presumably when you're observed, in the course of spousal death has nothing to do with your genetics. This is just a function of when the HRS happens to be scheduled to interview you. And so this really tells us that we're able to get some degree of prognostic power about who's going to experience the most pronounced level of depressive shock in the wake of spousal death if we're able to include genetics. And so that's potentially something that could be quite useful. A different example comes from a study that I worked on with Paige Harden, looking at the tracking of math courses that goes on in American secondary schools. What we have here is a river chart showing you the kind of trajectory of movement of students through the math course taking curricula, or the math curricula in high school. So in grade 9, you have most students starting in Algebra 1. The river is thickest here. Most of those students go into geometry. Some of them kind of just, remain in Algebra 1 or drop into a lower track. Most of the geometry students from 10th grade go into Algebra 2. You can see that by grade 12, most students have just stopped taking math entirely. And what I've done here is I've covered these different rivers by the genetics of the people in them. So basically, the darker the blue, the higher the mean polygenic score for a person in the river, where the polygenic score is designed to predict educational attainment. And what you would expect and what you see is that the kids that start higher and persist longer in math course taking are those that tend to have higher polygenic scores that are designed to predict educational attainment. Okay, so that's all well and good, but the interesting thing I think that we did here is we actually were able to use that to get a relatively nice observation that schools are treating children very differently. And so what we can do is we can look at how far kids go in math as a function of the school they're in when we hold genetics constant. And basically what we can see is that kids that have relatively lower polygenic scores tend to go a good bit farther in math if they go to higher status schools, right? And so this is a really nice signal that, schools are having, quite disparate effects on the kids they serve in a way that would be a little bit challenging to observe without the genetics. It's not that we, have no information that schools have effects. It's just that those effects can be challenging to observe given all of the different things that go into constructing the student body of a school. Okay, so to go back to the fact that genes are fixed at birth, I think this is really just an incredibly important point. They are the only thing that we can study about humans that are fixed at birth and unchanging throughout the life course. And this makes them very appealing as a potential tracer from which we can follow developmental processes. And as I have argued here, I think we can also use them to potentially identify differential responses to the environments, even when the environments may not be randomly assigned. There might be additional challenges we have to work with there, but I do think this offers a potential window of opportunity. Now, one complaint, I think, or one alternative suggestion you can make is, well, couldn't we just do this all with observed behaviors? And I raised this to note that, I think it's an important thing to think through. I think you potentially could, but you then have to start asking, which behaviors are relevant, which is not always, I think, an obvious question. Have they been measured at the appropriate times and with high fidelity? That those are non-trivial challenges. The behaviors themselves are frequently not exogenous. They're often on the developmental pathway you care about. And so I think genetics allow us to potentially circumvent all of these problems, given, again, the fact that they're fixed at birth and unchanging, whereas behavior is, well, let's just say it does not have that property. And so in summary, the point of opportunity that I think we have, and then I've assembled a bunch of other studies that I think also speak to this, is that genetics can be used as a potentially novel tool for studying change, whether that change is happening at the level of the society, as a few of these kind of indicate, or the level of the individual, which some of these talk about, or somewhere in between. And so I think this is an opportunity worth kind of taking seriously because it's worth at least thinking seriously about, because I think it's going to be something we see a lot more work on in the near future. Okay, so starting to turn from this point of opportunity to the potential challenges, I'm going to focus on a major challenge that has to do with our genetic predictor being confounded, but I want to acknowledge a second major challenge that is extremely important that I'm just not going to talk as much about today, and that's the fact that the polygenic scores and the genetic predictors that I'm discussing here are not yet ready to be deployed across all people, right? They are limited in their application at this point as a function of ancestry in ways that is quite challenging. And premature deployment of these scores, especially in clinical settings, may further exacerbate underlying health disparities, which is a situation we'd obviously just rather avoid. And so I'm not going to talk as much about these problems, not because they're not massively important, they are, but because I don't have as much novel to say about them. And I think we're in a situation where we're largely going to have to wait for the geneticists to kind of figure some of the technical issues out before much can be done. I think that work needs to be prioritized. It's incredibly important, and I want to acknowledge it as such, but I'm not going to speak a ton about it now. But if people have questions, we could perhaps come back to that later. The caveat and the challenge that I am going to talk about is the problem of social genetic effects. So this has been something that's been of big interest in recent years in this community. And so the underlying issue is as follows. What a GWAS would really like to do is to tell us about the association that goes basically between a person's genetics and some outcome for the person, some person level trait. It would like to tell us about the within person kind of biological impact of variation at a given point at a given point of genetic variation. So the GWASs, they want to focus on this black arrow here. However, there's a potential confounder in that if we have connection between the genomes of individuals and their social peers, and some role of influence from the genetics of this individual social peers and the individual's traits, right? This red path could be a confounding pathway. And this is a problem that people are starting to take quite seriously. And this turns out to be an old problem in the study of animals. So I actually learned about this when I reviewed a paper about mice. And the researchers there were interested in how the genetics of a mouse's cage mate may have implications for the phenotypes of the focal mouse. And I thought, well, this is pretty interesting. And so I started to think about and work on the kind of comparable problem amongst humans. In particular, I started to think not about the cage mate of a mouse, but the friends and classmates of adolescents in the AdHealth study. And so in this paper here, what we examined is whether there was a connection between the genomes of an individual and their social peers. We think there is. We also think that the genetics of an individual's social peers have some implications for an individual's outcome. So we both have evidence for this kind of red line and this red arrow. And this suggests that there might be some confounding going on if we try to interpret these GWAS findings as purely within person biological effects. They do not seem to be quite that simple. And so it seems to be the case that educational attainment-linked genetics that your social peers carry may matter for you, and this may have implications that I'm going to talk more about in a few minutes. But I want to say that it's not just educational attainment. And so Dalton has a very nice paper with some colleagues wherein they look at smoking-linked genetics of your grade peers and how they may also matter for your behavior, right? So the kind of picture that I've shown here is not really restricted to studying the genetics of educational attainment. This could pertain to any number of traits. This paper looked at educational attainment for the most part. This paper looked at something different. And so this is a kind of generic problem that we need to worry about. I looked at the connection between an individual's genetics and the genetics of their social peers, but there are other kinds of I looked at friends and classmates as one particular instantiation of a social peer, but there are other things you could fill in here. Augie Kong and colleagues asked the same question with respect to parents. And with respect to parents, you're in a very different situation because parents and their offspring, well, their genetics are clearly related, right? The parents are where the offspring get their genetics. And so you don't have to worry about this red line here. You know it is there. And so in this study, what they were able to show is that the genetics that parents don't transmit to their kid are still predictive of the children's outcomes in a way that, again, suggests the problem of confounding that I've been talking about. And this has come to be known as genetic nurture. And it's basically the notion that the genetics of a parent are predictive of the rearing environment of an offspring in ways that can confound simple interpretations of the effects of the offspring's genetics. And so this is something that's been undergoing a lot of work in the recent past. I wanted to note a few things that I think we're learning about it. So one thing we've learned is that you can triangulate with respect to this problem by using adoptees. And what you can do is you can use adoptees to note that adoptive parents and their genetics predict outcomes of adopted offsprings in ways that continue to suggest that the GWAS findings for educational attainment are not capturing something purely biological and within person. They're capturing something a good bit broader. This is a problem also, I think, of pronounced scale. Parental genetics are tied up with both prenatal conditions and very early levels of child achievement. And I think this set of findings in this paper suggests that this is just a really kind of messy problem that really is going to make it complicated to cleanly disentangle these things. And these challenges, I think, motivate a need for conceptual models for further informing us about what's going on. And so this paper down here is largely the work of Sam Trejo. And what this paper shows is that there are features of the world that we could set out to study that might offer some kind of further illumination as to what's going on with respect to the problem of genetic nurture. So I think this problem of genetic nurture is a very challenging one that we're going to need to better understand if we want to do real things with the findings from some GWAS. And so I think that's true. I also think that there is some very recent forward progress in this space meant to kind of resolve some of these issues. In particular, a big kind of GWAS within family study came out recently that I think is one step in the direction of resolving some of these problems. And so this problem of social genetic effects and the genetic nurture problem in particular, suggests that we need to be, I think, kind of realistic with ourselves when we go to interpret genetic association estimates. But it's also the case that other things are not immune from this basic problem. As I noted, parental genetics are highly associated with prenatal environments and other early life environment the child experiences. And so studies of the effects of early life environments may also need to consider parental genetics in the same way that studies of parental genetics may also have to kind of, be sensitive to the environmental stuff that the genetics are proxying for. And so this is a complex problem on both sides. Okay. So I now want to transition to the second part of the talk where I'm really going to focus on environments. So I've talked a little bit about environments thus far. I've talked about how they might serve as confounders, right? That's the problem of social genetic effects. There's also a classic interest in the possibilities that genetic effects may differ across environments. This is the idea of gene-environment interaction. And so now I want to talk about a specific point of confusion that is, I think, underappreciated when we talk about gene-environment interactions. And this is motivated by the observation that I made earlier that we have a massive amount of genetic data coming online. And as more and more polygenic scores are available, I think we're going to see ever increasing amounts of G/E research. And I think this is sensible. I can appreciate why it is we want to know the ways in which genetics effects may be contingent upon social environment. But I think we've really underappreciated the challenges associated with such studies. And that's what I'm going to talk about for the rest of the talk. I've grown greatly interested in these challenges. And in particular, I want to start by identifying a real source of tension between what we want from such studies and what I think it is we actually get quite often. So in this paper up here, we suggest a typology that I have found useful, and that is the difference between dimmers and lenses. So let me start with the lens. So we often, I think, in G/E studies want to think of environments as lenses. And what I mean by that is it's motivated by this notion of an optical lens. This lens reverses these two lines, right? It has this effect of reversing images if used to refract light. So, you have this red line on top over here, it passes through this kind of environment, and over here, the red line is now on bottom, right? So there's been a difference in, you have to fundamentally understand which environment you're in to understand whether this red line is going to be on the top or the bottom. And this is, I think, often the kind of theoretical idea people have in mind when they motivate G by E studies. They talk about genetic effects as being entirely environmentally contingent in a way suggested by this lens model. I think in practice, what we often get, or what I have come to call dimmers, the idea of being a light dimmer, right? You can kind of turn the light way down or way up. And what I have in mind is basically that environments are controlling the level of phenotypic variation that is being expressed. You can imagine some environments where phenotypic variation is very small, some environment where phenotypic variation is very big. And it is really those kinds of environments that we often are stuck studying as opposed to these kinds of environments. And I think this is especially important as you're seeing more and more work being done in longitudinal cohort studies of the kind that allow for the study of really big environments. So, birth year cohorts, right? That's a major environmental shock that might have this dimmer-like effect on certain kinds of outcomes. This really is in contrast to what you would often get in twin studies, where you have much smaller, much more micro environments that are really of interest. Now, I've tried to convince you a little bit that dimmers are irrelevant. What properties might a dimmer lead to? Well, the main thing that I think a dimmer would lead to is basically heteroscedasticity. So heteroscedasticity, right, is a problem associated with the error term. We typically, when doing statistical modeling, make a bunch of assumptions about the error term that are meant to make our lives easier. We assume that the error term is independent of predictors and that it has a nice normal distribution, but life could be much messier. These distributions could be asymmetric. They could have shapes that depend upon predictors, and they could otherwise just be ill-behaved. And what I'm showing you here is an outcome whose variation, the level of variation, the outcome really depends upon the environment, right? At this level, we have a lot of variation in the outcome. At this level, we have relatively little. And this is a problem of heteroscedasticity. And this is a problem that we may well have in certain G by E settings, right? So here I've kind of replaced that first figure with one wherein I have measured environment on the x-axis, some outcome on the y-axis, and I'm showing you three different levels of genotype, the red, blue, and green lines. You can kind of see this increasing level of predictive power for the higher polygenic scored folks in this sample. And you might look at this and think, oh, this is evidence of G by E. This is often the kind of thing that people present as evidence of G by E, this shifting level of slope from these three lines. And yet this scenario here has been generated entirely based on heteroscedasticity. There is no gene by environment interaction in the kind of classical sense, in the sense of just a product term between environment and gene being included in the model. This is all heteroscedasticity. And our concern is basically that a confusion about this might lead to kind of spurious findings of G by E that are really entirely due to heteroscedasticity or dimming. And so here's the scenario that I'm going to talk a little bit about. We're going to talk about a scenario wherein a genetic predictor predicts a constant proportion of the outcome variance, but we have an outcome whose variance shift as a function of the environment, right? That's going to be the heteroscedasticity, and the heteroscedasticity is basically going to induce G by E findings. So let me kind of walk you through this problem in a specific, let me kind of tell you a little about the methods, and then I'll tell you about the specific context. So this work I'm talking about now is all based on this preprint. And what we do in this paper is we contrast three different models, a kind of classic G by E model, right? That is the lens kind of model. Then a model that is really focused on changing levels of variation in the outcome, right? This is our dimmer model. And then we have a kind of flexible model that accounts for both of these. And what we did in this paper is designed an approach that basically allows us to kind of with some statistical testing, adjudicate between these three different models. And I want to note that I'm talking here about a specific G by E problem, but this is really a generic problem of heteroscedasticity that might be of interest in other settings, right? I'm not really talking about something that is restricted to the study of genes. And so the context we were interested in is one of BMI. And so let me tell you a little bit about why. So in the recent past, the GIANT consortium has led a large effort to map the genetics of BMI. And so we have, at this point, really good genetic predictors of body mass index. And several studies have examined shifting patterns of penetrance as a function of birth year cohorts. So they've basically shown that the polygenic scores for BMI have become more predictive in later birth cohorts. The biggest such study is this paper from JAMA a few years back. What they show here is across 4 birth cohorts, you see an increase in the steepness of the line, right? So the green line is the earliest birth cohort, the red line is the latest birth cohort, and you can clearly see the steepening of the lines. And my point is just that, this is exactly parallel to the figure I showed you a minute ago and might be driven by heteroscedasticity. And so we set out to kind of further interrogate that. I want to note that, this has been done by a few other groups, including one in which I was involved along with Dalton. And so this is really not meant as an attack. It's just me being continuously kind of intrigued by this problem and interested in thinking it through, again, from this different perspective. And the basic problem that I think we have in these studies is what I'm kind of illustrating here via chickens. It turns out that chickens have really exploded in size in the last 50 years. 50 years ago, if you went to the store and bought a broiler at the supermarket, you would have been buying a chicken that would have weighed about 1 kilogram, whereas now we can grow, let's just say, much bigger chickens. And in fact, humans have kind of done the same thing. That we've experienced a real shift in the distribution of our BMIs in the last 50 years. There's been something like a 30 to 60% increase in obesity between 1960 and 1994, which is roughly the years that I have in mind here. We can see this quite readily in the HRS data. So I'm showing you here I'm showing you QQ plots for BMI for similarly aged individuals. First when measured in wave 2, second when measured in wave 12, and you can see the wave 12 BMIs are just a good bit higher. Another way of looking at that is as a function of birth year. I'm now showing you BMIs by birth year, wherein the red line here is the 10th percentile, the top line here is the 90th percentile, and you can see this clear upward shift that might indicate heteroscedasticity. And so our concern is that this might drive the previously observed G/E findings. And so what we want to do is basically ask that question using our model that basically considers birth year to be a dimmer rather than a lens, right? And so what we're going to do is ask whether we have kind of a classic interaction effect or an effect that's better understood as this kind of scaling effect where the outcome variance is changing, but the genetics are playing the same basic role at all points in time. And to do that, I'm just going to show you a few pictures. The details are in the paper, so I'm going to kind of run through these quickly, especially as I'm getting close to time. But what I'm showing you in these pictures, here I'm showing just simulated data. And I simulated data first under the lens model and then under the dimmer model. And I'm showing you for different birth years on the x-axis, the amount of BMI-related variation explained by genetics on the y-axis. And under the lens model, we have this kind of this generic increase in the level of phenotypic variation explained by the genetics, whereas under the dimmer model, it's very flat, which is by design. That's what the dimmer is supposed to do. And so our question is, which of these is more consistent with what we see from the empirical data? Because this is all simulated based on parameter estimates obtained from the HRS data. And when we look at males, what we observe is the following. This red line is basically the pattern we get from the HRS males. And what we see is that this line is too flat to have come from a lens model wherein what you're really focusing on is changing levels of genetic explanation over time. What you have here is a situation wherein the level of BMI-related variation explained by the genetic predictor is really pretty flat. It's much flatter than what you get from lens models. In fact, it's very consistent with what you get from dimmer models, right? The flatness here is entirely consistent with the kind of flatness we get from our simulated dimmer data sets. And this suggests that what you need to understand what's going on with the males is a model wherein the outcome variance is changing, but the role of genetics is really constant. The story is a little bit messier for females. The data really aren't consistent with either of the simple forms. And ultimately, what we have kind of settled on is that if you analyze all respondents, you can kind of decompose this picture into one wherein the vast majority, about 2/3 of the apparent change in predictive power of the polygenic score over time has nothing to do with the kind of classic G by E effect, it's really due to changing levels of outcome variation. And only a third of that apparent change is due to kind of the G by E finding in the classic sense. And so at this point, I feel, like in the HRS at least, changing variation in BMI across birth year is really doing the lion's share of the work in driving the finding from this paper as well as the others. And that, so I think there's a lot of nuance in these earlier papers that is probably worth exploring on its own merit. But from this perspective, I think it's probably worth being very sensitive to the role of heteroscedasticity and measurement properties of BMI that had maybe not been considered as much in the past. Okay, so let me start to wrap up. So in this second half of the talk, I've described G by E as a dimmer, right? Wherein the key idea is that the environment is really controlling environmental variation. And I think this idea of a dimmer is likely to be of increased salience in the near future as we have genetic information in a large number of studies that have environments with really pronounced effects. And so I think we need to be increasingly sensitive to this kind of phenomenon. More and more, I've come to be concerned about the ways in which G by E studies maybe basically, they may be a good path for fooling ourselves and for constructing overly Baroque models to explain phenomena that can actually be explained by much simpler ones. I have a variety of papers that I could point you to that I think kind of help clarify what's going on here. But I think the fundamental issue is that interaction studies, that is studies that are looking at interaction terms, are really, they're of great conceptual interest, but they're really challenging analytically, especially if the outcome isn't well measured. They're almost designed to fool you if you're not incredibly careful. We have one preprint that basically shows that false discoveries are really, really high in studies of interactions. That's, I think, one way of interpreting the results from the previous slides. We have another study showing that if you're interested in something like years of education, you can really fool yourself by modeling it using the linear model. And all of this is just me trying to sound the alarm that G by E studies that aren't done very, very carefully could be producing way more heat than light. And I think that's a real concern given the amount of genetic information that's coming online. And kind of pulling back for a minute and thinking about genetic data more broadly, I think genetic data is just poised to play a really pronounced role in the next decade of the study of human behavior. I do think there are kind of reasons for thinking that such studies might be truly useful in helping us better understand behavior, but there are also ample reasons for care and caution. In particular, there are a lot of complex issues I haven't discussed. There are very thorny ethical problems in terms of how we might use them in, for example, IVF settings. Then there are also really complex problems when it comes to communicating these important scientific insights to the public. And I'm working under Daphne Marchenko here on a project that is meant to kind of better it's basically meant to systematize the way that such information is communicated to the public in an attempt to, hopefully improve public thinking and conversation about some of these issues. Okay, I'm going to stop there. The work I showed was, done in collaboration with a huge number of people. So thank you to all of them, and thank you to you all for having me.